Okay, it looks like the influx is slowing down a bit. I want to get started quickly because we have an outstanding group of scientists here today. This is actually, I'm a little biased, but this is my favorite session. I'm very much looking forward to this. So I want to do a very quick introduction of all of our speakers. And then what we're going to do is they're going to talk about their research. I want everyone to feel free to type your questions into the chat box, and we will do our best to address them as we go along. So on the panel today, we have Dr. Manju Kurian. She's a professor of neurogenetics at the University College London, Great Ormond Street Institute of Child Health. We have Dr. Ahad Rahim, a professor of translational neuroscience at University College London and the associate director for research in the School of Pharmacy there. We have Dr. Paul Kotzbauer, a professor of neurology at Washington University School of Medicine. Dr. Hugo Bellin, an HHMI investigator and professor of molecular and human genetics and neuroscience at Baylor College of Medicine. Dr. Guang Lin, a research specialist at Baylor College of Medicine and an expert in identifying novel molecular pathways in neurodegenerative diseases. And last but not least, Dr. Daniel Paul, Senior VP of Discovery and Platform Development at the New York Stem Cell Foundation Institute.
Okay, thanks, Erica. I'm going to tell you today about several treatment strategies we're pursuing to restore PLA2G6 enzyme function in INAD. And I want to thank the organizers for inviting me to talk to you today. All right, so a diagram of the PLA2G6 protein, which is produced from the PLA2G6 gene, and there are a variety of mutations identified in Susan Hayflick and colleagues' initial studies. Many of them just cause a single misspelling, 1 out of the 800 amino acids in the protein. And yet our impression is that this, and our data suggests that this causes loss of the ability of this protein to function properly as an enzyme. So I'm going to tell you a little bit about our work, kind of our early work in particular, about understanding this enzyme, how it functions, how we can measure it, which we think is very important for looking forward to treatment strategies as well. So what does it do? This is kind of a brief diagram, and for those who are not biochemists, you don't have to worry about the structures. But I'll explain that its job is to break down or break apart specific lipid molecules, convert one type of lipid molecule to another. And those are lipid molecules like phospholipids and lysophospholipids getting converted to fatty acids. And these lipids are important. The phospholipids and lysophospholipids are components of cell membranes. The fatty acids essentially can be used for energy, but also are kind of precursors for building these lipids that comprise the cell membranes surrounding the cells and compartments within the cells. And so we've looked in our lab, we can produce the PLA2G6 protein in its test tube and can introduce some of the mutations that are associated with INAD, and we can see how those mutations affect the function in different ways. We can measure the function of this enzyme in different ways. And so shown here is if we look at the wild type enzyme with no mutations, we can measure function, and for these mutations, we see very consistently a loss of function, and that's consistent with the genetics as well. And so that helps us think about what are the consequences of losing PLA2G6 enzyme function, and the function may be complex. There's evidence for what I would call homeostatic function, kind of regulating the balance of different lipid groups. Hugo Bellin is going to tell you about work he's doing looking at other function of the protein. So if it doesn't function, we're concerned about too much of the things that it would break down, and that may be consistent with the accumulation of lipid molecules we see in the neuroaxonal spheroids that essentially define INAD. Alternatively, things we can't see under the microscope are too little of the things PLA2G6 normally makes, and those would be the fatty acids that are needed for cellular energy and to make membranes. We've spent a lot of time working on a PLA2G6 knockout mouse model. This is a different model from what Ahad and Manju have been studying, and we think it's complementary in terms of the approach. This is a model generated by a targeted mutation, the traditional way of generating a knockout mouse model. It has a later onset of neurologic problems, but neurologic problems that we've learned to measure very precisely, and we've really optimized our ability to measure the neurologic deficits in these mice. In addition, we can see the neuroaxonal spheroids accumulate with age in the brain of these mice, and we have different ways of labeling them, and we can look at them in detail under the electron microscope as well. So with that kind of introduction of focusing on how we can study the enzyme and use the mouse model, I'll talk through several of our approaches to measure treatments, to develop treatments. And the first I'll talk about is gene therapy. So Ahad covered this very well in terms of an introduction, so I won't go into detail, but it is, in theory, a potentially excellent way to address the deficit. We can potentially completely compensate if we can get good delivery, completely compensate for the loss of function because of the mutation in the PLA2G6 gene. But as I said, the challenge is in delivery. And so we've focused on kind of measuring delivery. So if we build a vector containing this gene and deliver it to mice in different ways, how can we measure the delivery to assess just how good it is and to optimize it? And so this is one measurement we've been doing here, where we can take samples of the brain tissue or samples of other tissue like the liver in these mice. We can make measurements, and we can show that if we deliver these vectors, that in fact we do get protein production in the brain, and we can look at different variants and different vectors to kind of compare the delivery with this approach. Does gene therapy help INAD mice? So that's one of the questions we've been trying to answer, and I view these studies as complementary. These are complex questions, as Ahad nicely outlined, in terms of how do you best deliver it, how does the delivery change at different ages, and so we've been looking, in our initial studies actually at a more challenging situation in terms of delivery when the mice are 7 weeks or 11 months old, where the blood-brain barrier provides a significant challenge to IV delivery, and even intracerebral injections or intraventricular injections can be more challenging in terms of their ability to spread. And then we also worry about whether disease progression can limit the effectiveness of therapy. So we think these are important kind of complementary studies to really understand how this can work and how we can optimize this approach. So we are measuring the effectiveness with mouse behavior tests. And so far, our observations are that it's safe, but we haven't seen efficacy despite those measurements I showed you on the last slide that we delivered the protein. Now, we haven't jumped to the conclusion that you can't do it, you can't be successful. There are many things we're thinking about in terms of strategies to see if it can be more effective in this mouse model that we're studying. And those include changes in the vector, the delivery method, the dose, and even the use of different gene variants that exist for PLA2G6. I'm going to talk only briefly about enzyme replacement therapy because it's a strategy we've spent only a little bit of time on. But I'll talk about the idea is that it's not easy to deliver the protein itself. A lot of people ask about this, and I think that's where we are now. That's even more challenging to say we can deliver the protein, a purified protein preparation as a therapy. But what we thought about is ways we can adapt or modify the PLA2G6 protein that could make it better in a gene therapy vector if it could potentially be exchanged between cells. So we're doing some work on that. And then I'll spend a little bit of time talking about pharmacologic chaperone approach. And the idea here is that we could identify drugs that would improve the function of mutant PLA2G6 protein. When it has a misspelling, just one amino acid out of the 800, our understanding is that that impairs the ability of a protein to fold properly and form this very precise three-dimensional shape that it needs to be able to function as an enzyme. And there's data, good data for other proteins and other disorders that traditional drug-like molecules, by interacting with a protein, can stabilize and help a mutant protein fold better. And so we've spent some time looking kind of in early proof of concept experiments whether we could do that for PLA2G6. And although these don't immediately translate into therapy, they do support the idea that this strategy could work. ATP, a molecule that's used in the cells for energy, can't really be used as a drug. We find in our enzyme assays has the ability to improve the activity of both the wild type protein and a protein containing a mutation. And then other experiments we've done also support the idea that there are ways to take a mutant protein that's functioning very poorly and improve it, partially improve its function. Whether that's enough, we still need to, we still need to test that further, but it gives us some proof of concept. And we've actually started, have done a screen, developed assays to screen. In this case, we started with existing drugs to see if we could identify anything that would help the enzyme function better. We did not find any hits in our initial screen, even though we thought we had a very good assay and a very good, kind of measurements throughout this screen of 2,000 compounds. But we're thinking beyond that. We're thinking about whether we can further develop assays, including cell-based assays, that would allow us to probably more broadly look for compounds that may even have an indirect effect on its activity. And so, to summarize what I've gone through quickly, is that INAD is caused by loss of enzyme function of the PLA2G6 protein encoded by the gene. Our approaches are to use an INAD mouse model as well as enzyme measurements to develop strategies that would restore the function of the protein as a treatment for INAD. And so I talked about gene therapy, where you could deliver a replacement gene. And the challenge, I think, is delivery, and we need to utilize kind of these approaches for enzyme measurement and measurements in the mouse model to really see what it takes to optimize that, and eventually develop a treatment, what I think is a very promising avenue. And then I've talked a little bit about enzyme therapy, which I think actually, some approaches could be used to enhance the gene therapy approach. And same for pharmacological chaperones. I didn't touch on this, but we saw the improvement I showed in even the wild type enzyme with some treatments. And so even when you're not optimally delivering an enzyme, wild type enzyme, a replacement gene, if you can help that function better, that too could potentially boost an effect that you get from gene therapy. So we're thinking about these approaches perhaps in complementary ways as we move towards what we think is most promising, which is gene therapy. And then finally, I just want to acknowledge the people in my lab listed here, particularly the people in yellow who've worked on the INAD project, several collaborators, including Mark Sands, on our gene therapy work. And then I also want to acknowledge support from the NINDS, NBIA Disorders Association, and particularly supporting a strategy that I didn't talk about here, but I mentioned yesterday in the clinical talk. And then families and Ainsley's Angels that supported the Pharmacological Chaperone work, and then the INAD Cure Foundation supported gene therapy, as well as the Sweet Family Jasper Valentine Foundation and the Herschelman family. So thanks for your attention. If there's a little bit of time for questions, I'll be glad to answer.
Thank you so much, Dr. Kotzbauer. Looks like we've got a couple questions coming up. So maybe while Dr. Hugo Bellin gets his slides up, let's see. Well, these are to the panelists. So now I'm not sure if I'm supposed to ask the total group. Perhaps if those panelists are comfortable, they can unmute and ask. Okay. Well, perhaps we can get some questions from the audience in a bit. Apologies for that bit of confusion. Definitely keep the questions coming. Everybody's going to be sticking around, so we'll do our best to cover everybody. Thank you so much, Dr. Kotzbauer. We really appreciate your time. So next we have Dr. Bellin. If he can bring his slides up.
I've been trying to share my video, but it doesn't seem to work. The host has stopped it.
Oh.
All right. Now
there we go.
Can you see my screen?
Yes.
All right. Huang and I decided to make this one presentation so we could spend 20 minutes covering a little bit more ground, and I'll be presenting Huang's work. So the main question is, how does loss of phospholipase A2G6 affect neuronal health, and what are the mechanisms of action? And given that we work with fruit flies and most of the patients and parents of patients are not familiar with fruit flies, that briefly introduce the fruit flies and why they're being useful. So flies have 16,000 genes versus human 24,000, and 70% of the genes are conserved. And they have a short lifespan, they're cheap, they're easy to breed, and they have the most sophisticated genetics of any animal species, and therefore allow you to do really sophisticated experiments. They also have a pretty complex nervous system. People think that the fly nervous system is really simple, but it is not. In fact, it has as many different neuronal population as a human brain. The main difference is that there's just many fewer of each of those neurons. And so we've been working with fruit flies, and I just wanted to briefly highlight how they've been successful in helping diagnosis of undiagnosed diseases. We are part of the Undiagnosed Diseases Network, and about 5,000 patients have so far applied. And they can register at the coordinating center, and then they can get a workup in any of those 12 clinical sites where typically they sequence the patients and the parents, and they find variants in genes that might be the cause of the disease, and they submit them to the model organism screening center that I had at Baylor College of Medicine. And we developed sophisticated informatic tools to really mine all the data in the literature and come up with the best variants and the best genes that we should prioritize. And we work with 3 model organisms, worms, flies, and zebrafish, to try to help in the diagnosis and unravel the molecular mechanisms that are the cause of these disease. And in the last 4 years, we've been quite successful. And as you can see here, we've participated and helped in the diagnosis of using fruit flies of quite a few genes that are listed here in their publications. In many cases, we can further the studies and really dig into mechanisms. And once we understand the mechanisms, we can come up with drugs. And I wanted to say that, for example, here, this drug, rapamycin, is now being used in children with mutations in OGDHL. In the case of ACOX-1, we've also worked out the mechanisms, and we now have 3 drugs that have been shown to be efficient. Same for RF2-BPL, and I wanted to briefly talk about phospholipase A2G6, since that's the main topic of this and the drugs that we found so far to be efficient. So a few words about the fruit fly. When this gene, there's only one gene, and it's called phospholipase A2V6A. In flies, the flies have a short lifespan. They start dying from day 15 on, and by day 30, most of the flies are dead. Normal flies live for 70 or 90 days. They have an increased sensitivity to starvation. They have olfactory dysfunction. They have visual defects. They have severe climbing defects. And they have strong increased bang sensitivity, which is a measure for seizures. So you can bang the flies, and many of the flies will start seizing. And this is a very sensitive and easy assay. And as you've heard, the patients also have seizures. And they progressively lose the ability to see, because we can measure that with electroretinograms or ERGs. So what about the cell biology? I briefly wanted to introduce some of the cell biology and the organelles that are important here in the cell. So I'll come back to this, but the endoplasmatic reticulum is where proteins and lipids are synthesized. And the key lipid that I'll be talking about is ceramide. And ceramide is then transferred to the Golgi. And in the Golgi, the ceramide is packaged in vesicles that are secreted and put into the plasma membrane. And what ceramide does is it stiffens the plasma membrane. So it provides a backbone to the plasma membrane. But like all proteins and lipids that are present in the plasma membrane, they're endocytosed through an endosome. And the endosome that carries these proteins and these lipids then travels to the lysosome, where these lipids and proteins are degraded. I'll briefly talk about this little organelle, the retromer, which is an organelle that essentially pinches off vesicles from the endosome to recycle lipids and proteins back to the plasma membrane. And so our model that we worked out relates to the endosome, the lysosome, but also to another organelle, the mitochondria. And I'll come back to that in a minute. So the loss of phospholipase A2C6 causes an increase in an enlargement of the lysosome, and that's documented here with lysotracker. The lysosome can be thought of as the stomach of the cell, which digests, as I said, the lipids and the protein. And you can see that there is an expansion of the size and the number of lysosomes, maybe not that much the number, but certainly the size. And that was verified with transmission electron microscopy in fruit flies, showing that over time we see an increase in the number of lysosomes based on electron microscopy data. We also see these tubulovesicular structures that Paul just talked about. The next organelle where we see a defect is the mitochondrial architecture. And you can see that the mitochondrial architecture over time falls apart, in that between day 15 and day 30, a very significant number of the mitochondria become abnormal and have these abnormal structures that are shown here. There's also a problem with the dynamics of the mitochondria when this occurs. Third, what we see is that there's an accumulation of glucosal ceramide. And that is shown here. When we lose phospholipase A2G6, you can clearly see that this antibody that recognizes glucosal ceramide is quite dramatically elevated. And this is in the visual system in this particular case. And this phenotype is actually mimicked by proteins that are part of the retromere. And I'll come back to that later, because phospholipase A2G6 binds these two proteins and is required for the function of these two proteins. And so they all cause a very similar phenotype, which is an accumulation of glucosal ceramide in the cytoplasm of these neurons. So in brief, what we've shown before is that ceramide is synthesized, we know, in the ER, is transferred to the Golgi, as already mentioned, and is endocytosed from the plasma membrane. It turns out that phospholipase A2G6 binds to VPS35 and VPS26, which is interesting because VPS35 causes Parkinson's disease. And so we argue that when we lose phospholipase A2G6, we lose these two proteins, and that impairs the function of the retromere. And therefore, there's no rescue pathway for proteins and lipids that is functioning normally here. Therefore, there's too much lysosomal trafficking. There's an expansion of the lysosome. And as I've shown you, there is an expansion of the lysosome. And in the lysosome, ceramide, through an acidic sphingomyelinase, is degraded to ceramide. And ceramide will be elevated in the cells, and we've shown that ceramide is elevated, and starts integrating in membranes and stiffen up the membranes of the cell and the plasma membrane. And so it makes the cell kind of stiff and old in a way. The cypramine is a drug that inhibits acidic sphingomyelinase in the lysosome and has clear beneficial effect. It doesn't rescue the phenotype, but it clearly has a positive effect. I now want to turn to cells that are derived from patients, from fibroblasts, and look at what's happening when we culture dopaminergic neurons that are derived from these cells. So this is a collaboration with the New York Stem Cell Foundation. And physicians collect fibroblasts and lymphoblasts, and they differentiate them back into iPSCs. So they differentiate the fibroblasts into iPSCs, and those can then be stored. And then they taught us how to produce NPCs, neuronal progenitor cells, from these. And then we can take these neuronal progenitor cells and differentiate them into dopaminergic neurons. And this allows us now to look at lysosomes, at mitochondria, and at glucosal ceramide and ceramides in these cells of these patients. And you've heard that we need, ways to be able to assess if a drug works, and that we need biomarkers. And so what we can do is, in these cells, we can look at the proteins, the levels of the proteins from the retromere. We can see if there is lysosomal stress. We can look at mitochondrial function, and obviously we can then test these drugs in these cells that we've already tested in fruit flies. And so the key feature is that what the New York Stem Cell Foundation did was to take these patient cells and corrected the mutation in the INAD part 14 gene to make it a wild type gene, a normal gene again. So we have a perfect control because we have the cells that like the protein and the cells that are corrected and have a wild type protein. And we started looking at it, Wang started looking at these. And we started looking at the features that we all saw in the fruit fly. And so these are the edited cells, the iPSCs, the edited cells. They're wild type for phospholipase A2G6. And these are the uncorrected cells, or patient cells that have not been corrected. And you can see that there is a dramatic increase in the number of lysosomes, especially again, the size of the lysosomes in these cells. So this fits with what we've seen in the fruit fly. Now we can look at the mitochondria and look at the mitochondria of the cells that are normally, that are corrected. And you can see there is a network of mitochondria. These are the organelles that produce the energy for the cell. And when we look in the patient cells, we see a very different profile. We see that the mitochondria are not as well connected and don't form a network and have a very different morphology. So this again is in sync with what we've seen in the fruit fly. Third, we can look at glucosylceramide levels, which we've argued that ceramides are elevated and then glucosylceramide is elevated. And as you can see here, we took the iPSCs and differentiated them in dopaminergic neurons, which we've shown, and I'll show you that in a minute, accumulate high levels of ceramides and in the mouse. And you can see here that, again, there is a pretty significant increase in glucosylceramide in the cytoplasm of these dopaminergic neurons that are differentiated from these iPSCs. So when we look at the mice, we also see that in these mice, in the wild type mice, in two organelles, the cerebellum, which you've heard is affected, and the dopaminergic neuron, we see a pretty significant increase in glucosylceramide in the dopaminergic neurons and in the Purkinje cells, in the mouse that are these knockouts that Paul just talked about at 13 months of age. So we argue that whatever we've seen in fruit flies is evolutionarily conserved, is present in the cells of the patients, and is also observable in these mice. So for the patient cells, what I've shown you is that there is an expansion of lysosomes, there is an aberrant morphology and dynamics of the mitochondria, and there is an accumulation of glucosylceramide, which is very similar to what is observed in flies. Therefore, drugs that work in flies may work in patients. So we use an INAD fly model to test and screen drugs that alleviate neurodegenerative phenotypes. And the way we do this is we use a bank sensitivity assay. I told you that the bank sensitivity assay is an assay where we take the flies, we shake them, and when we shake them, and if they're susceptible to seizure, they will show this seizure phenotypes. It's kind of a trigger to promote seizures in flies that are not healthy. And when we do that, you can see that the flies fall on their back and they start seizing. And this is very pronounced in this fly model. And so now we can, when the flies eat close, we can put them on a drug, put it in the food, they will take up the drug, and we can measure the time that they are seizing or paralyzed. So we've done this for a series of drugs that would predict to change the acidification or suppress the acidification in the lysosomes. And as you can see, hydroxychloroquine, which promotes or elevates the pH in the lysosome and therefore should disrupt the function of the lysosome, sees much longer than the animals that are that have lost a mutation. So when we test these animals with hydroxychloroquine, they cease for much longer. And you can now look at all these drugs that we've tested here, and you notice that ambroxol suppresses the seizures, that genistein also suppresses the seizure, that a drug that has been shown to act on the lysosome also suppresses seizures, and another drug that acts on lysosome also suppresses seizures. So all of these drugs here that are shown here in a dose-dependent fashion suppress the recovery time that is needed, that is present in the wild type. So this then now allows us to go back to the cells from the patients that we have and check if we can suppress the lysosomal expansion, the aberrant mitochondrial defect, the increase in glucosal ceramide, and test these in the dish. We've not done this yet because we need to expand these cells, and we're working with the New York Stem Cell Facility to try to expand these cells and to start testing these drugs that we've shown are working in fruit flies. We can then test them in mice if we can show in a tissue culture system. I want to end and draw attention to the fact that there is an increase in glucosal ceramide. Glucosal ceramide is a major player in a disease called Gaucher disease. And in Gaucher disease, there is an enzyme that takes the glucosal ceramide and turns it into ceramide. And you can see that ceramide is central to a whole series of diseases that have ceramide or modifications of ceramide, like in Krabbe disease, where galactosyl ceramide is hydrolyzed to ceramide. In Gaucher disease, glucosal ceramide is hydrolyzed to ceramide. And all of these diseases are devastating early onset childhood diseases. And we think that glucosal ceramide, in this particular case, and ceramides, because other ceramides are also elevated, play a critical role in the progression. But it's also interesting to note that many of these players are also playing a role in Parkinson's disease. And we tested what happened in flies with a knockout of GBA, and we noted that there was a severe accumulation of glucosal ceramide. So this is a wild type control. These are the photoreceptor neurons. These are the glia. And you can see here that in contrast to what we saw for phospholipase A2G6, in Gaucher disease, there is an accumulation of glucosal ceramide in the glial cells. And this leads to a very rapid neurodegeneration. This accumulation of glucosal ceramide in the cytoplasm of the neurons, as well as in the glial cells, leads upon light to very rapid neurodegeneration. It leads to real mayhem in the eye when we expose the flies to high levels of light. And so I think this is important because this gives us a cue that some of the phenotypes that we may see or are associated with INAD may also relate to this phenotype in Gaucher disease. And I'll end here and be glad to take questions. Guanglin is the person who did the majority of the work. We are very grateful to INAD Cure, where Lina Panwala and Amanda Hope have really helped us with this work. And we thank HHMI for all their support during the years as well. Thank you.
Thank you so much, Dr. Bellin. That was wonderful. We've got an active chat going. We've also been, we're just about 5 minutes until the official time, but I've gotten permission to go over time. So we definitely have time for Dr. Paul's presentation. I'd say let's move on to Dr. Paul's presentation and then make sure you answer questions in the chat. And hopefully maybe we'll have a couple minutes at the end for some more questions. Thank you again so much, Dr. Bellin.
Thank you.
Okay, so let me share my screen. Okay. Alrighty. So again, thank you for having me here today. My name is Daniel. I'm from the New York Stem Cell Foundation that Dr. Bellin was just referencing. And I'll touch on some sort of the parallel work and some of the work we're doing here as well in this space. So just as a way to set the stage, just to sort of expand upon what an induced pluripotent stem cell is and iPSC. It's a technique that we've used now for over a decade and allows us to take a sample from a patient or a healthy donor, anybody you like. And it can be a tube of blood, just like going to the doctor. It could be a set of cells that we obtained from a skin biopsy. The skin biopsy is very small, maybe the size of an apple seed, and we can grow out cells from that, these skin cells. By overexpressing a small number of genes, these are called the Yamanaka factors, and I'll explain why in a second, we can revert these into something called an induced pluripotent stem cell. I'm just going to turn my spotlight on here. So that's the cell here. These cells are effectively indefinitely expandable. We can just keep growing and growing them. And most importantly, we can convert these into really any cell type of interest. And obviously, where relevant, a disease-relevant cell type. And so this technology stems from around 2007, 2008. And the importance of it was identified very quickly and has already led to a Nobel Prize for its inventor, Shinya Yamanaka. And so this bypasses a lot of the issues surrounding, obviously, embryonic stem cells and really allows us to make these patient-specific stem cells. And so just to illustrate that with some graphics, this is what your skin looks like if you magnify it. Here we've got it stained in particular colors, but these are your fibroblasts, what they look like. And if we overexpress these genes, we effectively turn them back in time into these cells here, and these are stem cells. And so every dot that you're seeing here, every circle, if you like, is the nucleus of a stem cell, and they grow in these colony-like shapes, is what we call them here in the lab. And it's these cells that if we give the right instructions, and the instructions come in form of the liquid that we feed these cells, we can turn them into any cell type that we like. So here, for example, the cardiomyocytes, and hopefully you can see this beating away in the dish here. Or in the case of INAD, a particularly more relevant cell type, as Dr. Belen was saying, dopamine neurons. So here you can see dopaminergic neurons that have been grown from those same kinds of stem cells. And in theory, these cells allow us to make any cell type in the body. So in the case of a neurological disease, obviously a neuron, but it could be a liver cell, it could be a kidney cell. And importantly, you can envision a day when if you do have a drug, not only could you test it on the relevant cell type, but you could look for off-target effects in, say, a liver cell and maybe early on identify potential toxicity-related events and things like that. So here at the New York Stem Cell Foundation, we are very interested in a wide range of different diseases, particularly neurological, where we work on Alzheimer's and Parkinson's and multiple sclerosis. But we also have a team doing bone development. We have a team that work on diabetes, and really across the board, we have researchers working in different spaces. But it is very challenging work. It takes a lot of effort. And so over the past decade, we've also put in place the ability to do this at scale. And so we've brought together 3 typically distinct disciplines of hardware engineering, computer science, and biology to develop a platform here that allows us to do stem cell research at sort of an unprecedented scale. So we've harnessed the power of robotics and various associated technologies. We've built sort of software backend so we can track everything, as well as building out data storage, data analysis capabilities here. I'll come back to machine learning in a few moments. And obviously the biology underpins most of what we do. And what that looks like in practice is something like this. So we have these robotic liquid handlers that can do exactly what I would do by hand, picking up plates, you can see some of the liquid there. The liquid keeps the cells healthy, just like you and I need to eat, so do these cells. And these things run 24/7. When we're sleeping in the evening, the microscopes are taking images of the cells, and then when they're not being fed or being processed, they live inside this incubator here, keeping the cells at the appropriate temperature and things like that. And this particular image that we're seeing here has lots of circles on top of it, and each circle represents a different position in the plate. And so I could have a different person in each position, or I could put a different drug in each place if I'm doing a drug test, and I'll come back to that in a second. And so our work in INAD is relatively recent, and but it's growing very, very quickly. And there are 2 things that we've been looking to do, particularly with INAD cure, but it's a growing interest sort of in this space. We have a big interest in rare disease. So one, the goal was to develop a biobank, so to create a community resource of patient cell lines, so that is skin or blood cells, as well as iPSCs derived from those cells, that could be shared, globally to advance INAD research. As importantly as getting patient cell lines is getting siblings, if possible, or familial relatives, just as important as the patient samples. And then the second goal is to use our expertise in this sort of automation and stem cell biology to see if we can develop new models or new approaches for high throughput drug screening. And so this is what the repository currently looks like. To summarize it, it sort of has 15 fibroblast lines so far. 6 of those are iPSCs, and I'll come on to isogenic controls and what that means in a second. And this expands, this is in addition to the many thousands of samples that we've collected along the way for a whole raft of different diseases. And so where we need additional controls, we can tap into our expanded repository. And this can collect everything from medical information to information about the samples, and it's all de-identified so that we can do this research without sort of exposing any identities. And iPSCs have, as Dr. Belen was saying, have a particular capability in that we can alter them. So if you imagine, as was being discussed, that there are various mutations and there are many hundreds that have been associated with INAD, as there is with any disease. What that means in practicality is that in your DNA, where you would normally have these 4 letters, let's say, for example, in most of us, this would be an AT relationship here at this position. Someone with a disease might have, for example, a GC, and it's this change in the DNA that is driving the pathogenesis of the disease. And so what we can do is take effectively a pair of biological scissors, and we can cut a portion of the DNA out and replace it so that once again it has the correct genetic sequence in that position. And this technology you've probably largely heard of. It's called Crispr. This particular enzyme here is called Cas9, and it has a very specific way, I'm not going to go into the technical details behind it, but a very specific way of going into the right location in the DNA, and it has these scissors built into it. So that's underway at the moment, and as was shown, we're working actively with other researchers to enable these tools to further studies. But with many hundreds of different mutations, it will be important to see whether it is a one-fit-all solution for any one drug that ultimately gets identified. So this Cas9 solution, this CRISPR-Cas9, allows us to repair these mutations. That could obviously potentially one day have implications in cell therapy as well. And so comparing cells with and without the mutation, as Dr. Belen was showing, we can then look for effects of the disease in cell types such as dopamine neurons. And these tools are extremely valuable in drug discovery. But there's 2 important questions when it comes to drug discovery. One is, what does a drug need to do, and where in the cell should we be targeting? As you just heard, there are many associated features of a cell that seem to be affected in this disease. And so what is the target? And maybe the question is there even one single target? So it could be the mitochondria that are the energy provider of the cell. It could be the endoplasmic reticulum where proteins are made, or it could be the Golgi, the FedEx, if you like, of the cell where they're packaged up and moved around. So, typical drug discovery would look a little bit like, it can look a little bit like this. You would put cells into a dish like this. Every little circle here would have some cells inside it, and a new drug would be applied to each and every well one by one. And so that's fine. We put this in, we add the different drug. And typically in drug discovery, we're looking for a single hit. So we're looking for something that changes. And here, for example, it could be that these 3 things now happen to glow green because of how we're measuring it. And that would constitute a hit. But what about all these other wells? Does that mean the drug doesn't work? Or does it mean that it just didn't work in the way we're measuring it here? And that's a really important distinction. So the question becomes, what if we're measuring the wrong thing? It's very hard to measure lots of things at the same time. When we're doing an assay, we're typically just looking at one feature, say the lysosome, or we're looking at one thing, the mitochondria. But again, it increases the chance that maybe you will miss something. So this is where we're switching to using machine learning here to empower that. And what do I mean by machine learning? Most of you will be familiar with something like the Google Assistant or Siri on your phone. That's a great example of where machine learning in everyday life has just become routine for us. It could be in self-driving cars, where we think about one day maybe we will have autonomous cars and it has to identify all these different things. And so these technologies have come a long way where in real time we can identify things like this. So it only makes sense that such technology would have a use in human biology. And what we're able to do now is build upon work that others have started, but effectively apply different dyes to stain lots of things within the cell at the same time. So here we've got the nucleus for the DNA, we've got the endoplasmic reticulum, we've got an RNA stain, we've got the cell morphology with actin, and we've got the energy centers of the cell with the mitochondria. And if we do this on enough cells, we can start to build what we call a phenotypic profile. We aren't just looking at one thing, we're looking at all these different things. By eye, I can't process the subtle changes that might be in any one of these things, but with a neural network and machine learning, we can start to do that. And so it allows us to start building these profiles of what the cell looks like. And so I'm almost done here. If we take an unbiased approach to machine learning, what that means is that we can take cells from healthy individuals and cells from patients. It could be neurons, it could be skin cells, it could really be any type of cell. We take enough images and enough photographs of these cells. It could be the corrected ones that we've made with Crispr, really any kind of cell that we want to do. And with high throughput automation, we can take lots and lots of these pictures and we feed that into a model and we start to train a model. And so eventually it might be able to predict which photographs come from healthy people and which photographs have come from patients with a disease. And that obviously means then if you apply a drug, you'd be able to see which ones start to look healthier again. And it asks the question, are there drugs out there right now that are already approved for different conditions that could be repurposed? And the only reason maybe we've missed it so far is that the test we were using just didn't pick up on the change. It wasn't the one thing we had to be looking at that point in time. And so this might sound quite fancy, but actually we've already had good demonstration that this is possible. So if we go back to our skin cells right at the beginning, these skin cells that seemingly have very little to do with a disease like INAD or a disease like Parkinson's disease, well, actually we've been able to distinguish Parkinson's patients from healthy patients just by looking at these skin cells. And so this is quite a busy slide, so and I have very limited time, but effectively we went to our repository where we've collected many hundreds of samples and we sort of made populations of people. We had roughly equal populations of healthy people. It's about 100 people or so in total here. Half healthy, half with Parkinson's disease. And some of them had a very specific form of Parkinson's disease. We put them through our automated processes, took thousands and thousands of these images, in fact, millions of these images, and put them into a neural network. We had the machine start to train it, and it was able to very, very accurately start to classify this is a Parkinson's patient and this is a healthy patient. And even more, it could subcategorize the different mutations in this as well. And so we're building on this now, and this is just at the starting point. So I'm hopefully able to tell you again in the future a little bit more about the results that we're getting, but we're very, very excited about what we're seeing with INAD. And so we think stem cells, they're going to be the future of medicine in one way or another, whether it's in drug discovery, in cell replacement therapy, or just helping our understanding of disease research. One of the things is we do absolutely need more and more samples over time. So Lena and INAD and the team there have been fantastic in helping us connect with families and bring them into the repository, but do feel free to reach out to the foundation or to us directly, and we would happily love to talk more about whether we can bring your samples in to the study. As everyone has said, I'm just one person, part of a huge giant team of scientists, engineers, data scientists, you name it, they're all here, and incredibly thankful for this team. That is it for me.
Thank you so much, Dr. Paul. I really appreciate you being brave enough to take the anchor leg on a session that's running over. We've just heard incredible science from a number of scientists all over the world. There's a lot of information coming at everybody. The chat box has been blowing up. I would say we've got just a couple of minutes, and I think rather than going hardcore scientific on the last question, I really liked this from Mariana. She asked, what can parents do, or maybe families in general do, to help with the research? So Dr. Kurian answered about being involved in some of the natural history studies. Dr. Paul, you talked about getting tissues into your repository. Maybe some other people on the panel can give brief comments about what families can do to be more involved with the research.
I'm still, I can't stress enough the importance of patient samples. The more we have, the more we can check whether something that we're finding is a one-off, or does it represent something that's really significant across a wider body of patients. There's questions about how to get involved. I think, as I said, reach out to the INAD Cure Foundation or to our website and to do. But yeah, the more involvement from patients, the better it is.
I was only going to share one thing that I've also been involved in gene therapy for AADC deficiency, where the families have really driven the development of that. The one thing I worry about, I know there's lots of families, and that's why I want to say this right now to all you, all the families out there. I mean, as I said, I'm a clinician through and through, really, and look after many, many families with INAD, like the families on this call. One thing I worry about with INAD families is that the regression can be so fast that I feel a lot of families lose hope. And I really want to emphasize today that I hope you feel, having heard all these talks today, that there is hope, that we are all working very hard between gene therapy, enzyme replacement therapy, high throughput screening, IPS models, that really research is accelerating, but we need your support to bring it to the next stage, as it has done for many other conditions like AADC deficiency, Batten's disease. I think we're in that frame. We need your support. So exactly like people like Dr. Paul has said, things like skin samples, blood samples, urine samples, participating in awareness, all these things are really important, because we are your advocates to get this, all these things help funding, help long-term kind of translation, so it is really important.
Any last comments from the other panelists? Otherwise, we can wrap it up so the next session can get prepared and ready for their webinar. Thank you again to everyone. This was outstanding. I learned a lot. I loved it. I hope the families on the webinar got to learn a lot today too. And I'm sure all of these scientists would be happy to hear from you. So email, ask questions along the way. Thank you all for a wonderful session. Have a great day.
Thank you.
Thank you.